Hi, my name's Erta and this is
Dr. Toti.
and today we will be exploring gene expression and the role of stem cells in medicine today.
Hope you enjoy the video.
Bye!
Lactose not present. E. coli produces the enzyme degalactosidase to break down lactose, which is a carbohydrate, into galactose and glucose, which are monosaccharides. The three structural genes, LAC-Z, LAC-Y, and LAC-A, produce proteins that help bacteria digest lactose. When lactose is not present, this is what happens. The regulator gene produces a LAC repressor, which binds to the operator site when no lactose is present. This blocks the RNA polymerase, preventing it from binding to the DNA. When lactose is present, it binds to the repressor, changing the repressor's shape so it can no longer bind to the operator site. RNA polymerase can now begin transcription of the structural genes. Transcription in eukaryotes. First, RNA polymerase binds to a section of the DNA adjacent to the gene to be transcribed, called the promoter region. Only when the enzyme attaches to the DNA needed will transcription proceed. However, the gene will remain switched off until a regulator protein attaches to the site also. How can transcription be prevented? A protein repressor can attach to the DNA of the promoter region, which blocks the attachment site. A protein repressor molecules can attach to the regulator proteins, which prevents them attaching. In both these cases, the gene is switched off.
Following on from our explanation of gene expression, we can now develop our understanding of stem cells and their role in medicine. Here we have Dr. Toti, all the way from Potent, USA. Thank you for joining us, Doctor. So, Doctor, what are stem cells?
Well, Erta, that's an excellent question. Stem cells are found in our bodies, and they are unspecialized cells which can turn into different types of cells.
Really interesting. So, are there different types of stem cells?
Well, Erta, there are. There are three kinds, you see. Totipotent, which are found in embryos, and they can turn into any kind of cell. Pluripotent, which are also found in embryos, they can turn into most kinds of cells. And multipotent, which are found in adult bone marrow, and they can only turn into some kinds of cells.
So why do we have different types of stem cells in our embryos?
The 8-cell stage, our cells are all totipotent, as they've undergone 3 complete cell cycles. Therefore, the zygote is ready to turn into a human. However, in order to do this, we have to be able to make all kinds of cells. Tissue, organs, everything. However, 5 days after conception, it forms a ball of cells called a blastocyst. And the innermost cells goes on to form tissues of the developing embryo. And it's made of 50 cells, and all of these 50 cells are pluripotent because it can only form tissues of the body.
So, what are multipotent stem cells?
Well, Erta, multipotent cells are adult stem cells. Now, they're found in the bone marrow, and these are needed because we have many types of the same cell. For example, blood cells. Now, they could be red blood cells, different kinds of white blood cells, or even platelets. Now, all 3 of these are needed for thermoregulation, transporting nutrients and waste around the body, and protection of the body.
Interesting, thank you, Doctor. I wasn't aware of how many different types of cells we had in our body. Is it the same thing for plants?
Actually, no. You see, many plant cells remain totipotent throughout their lives. Unlike in animal cells, plant cells can de-differentiate and turn into completely new cells. This allows gardeners to regenerate whole clones of a plant using only a stem, or a leaf cutting, or a root.
So linking back to the previous idea of gene expression, how do stem cells link?
Well, so stem cells become specialised through differential gene expressions. So the DNA of a cell, the genes are either switched on or off, and a different combination of this then creates new and different kinds of cells.
Oh yes, that makes sense. So, what are some current uses of stem cells in medicine today?
Well, currently we are studying leukemia using stem cells. Now, leukemia is where it's a cancer of the blood, and it kills all of the stem cells in your bone marrow. Now, We're trying to transplant new bone marrow into the patient, and then they have new stem cells, which is treatment for leukemia.
So what kind of other research are you doing to the use of stem cells?
We're currently doing quite a lot of research into stem cells, but the main two that we're focusing on are spinal cord injuries, and so we're trying to replace damaged nerve tissue, and this could potentially cure paralysis. The second one is for damaged heart tissue, for example from heart attacks, and we're trying to replace these damaged tissues using stem cells.